Hello, I am Shaili Dash. Welcome to the Knowledge Club Online. Today, I am going to discuss about viral replication cycle. Due to lack of biosynthetic enzymes, viruses utilize the biochemical machinery of the host cell to synthesize virus-specific macromolecules required for the production of virus protein. We know that the genetic information is contained in the viral nucleic acid. The general outline of the replication cycle is similar in most viruses. The long incubation times of many human virus diseases indicate that they replicate slowly in host cells. In tissue culture system, it has been shown that most human viruses take from 4 to 24 hours to complete a single replication cycle. In general terms, 6 main stages can be recognized in the multiplication of viruses. Number 1, attachment or adsorption. Number 2, penetration. Number 3, uncoating. Number 4, biosynthesis of viral proteins and replication of viral nucleic acids. Number 5, assembly. And number 6, release of progeny viruses. One by one, we will go through these 6 processes. Take a look. In this video, I will discuss on first 3 stages of replication and one part of stage 4. Rest of the parts of the replication I will discuss at our next video. So for gather total knowledge on viral replication, you have to watch and follow both videos up to the last. So firstly, I am starting with attachment or adsorption stage. The viruses come in contact with cells by random collision. But attachment depends on the recognition of target cell and presence of specific receptors on the cell surface. The first step is interaction between the virus and the specific receptor site of host cell, usually glycoproteins, the specific proteins on the surface of virus particles, on the plasma membrane resulting in its attachment. In example, we can memorize our previous lecture viral proteins, hemoglobin proteins of influenza viruses. If you missed that lecture, please visit our channel or click at the I button top of this video for getting that link. Host, each cell can have many receptors which near about 5 lakhs. Viruses make use of a variety of membrane glycoproteins as their receptors. The primary functions of the cellular receptor molecules are not related to their role as viral attachment sites. Some being membrane permeases of molecules or hormone receptors. Different viruses may use different receptors. The infection is multiple in most cases. Hundreds of variants of the same type absorbs to the cell. However, once infected by a given virus, the cell becomes refractory to additional variants. Some of the best characterized receptors present on cell membranes are Epstein-Barr's virus, which target cell is B cell, and the receptor is C3 complement receptor, which called CR2-CD21, and the HIV virus, the target cell is helper T cell and the receptor is CD4 molecule. Rhinovirus, it's attached with the epithelial cells and the receptors, the intracellular adhesion molecule 1, ICAM1, and poliovirus epithelial cells, immunoglobulin superantigen family protein is the receptor. Now going to the second stage, penetration. Multiple interactions between the viral attachment proteins VAPs learned at the previous lecture initiates the internalization of the virus into the target cell. The mechanism of penetration or internalization depends on the structure of virus and the cell types. There are 2 general mechanisms. The number 1 mechanism is the membrane fusion. The membrane fusion mainly happen at the enveloped viruses like paramyxovirus, retrovirus, etc. Fuse their membranes with acidification after attachment of the virus to the host cell receptor. Acidification generates conformational changes in the virus coat, which can release the virus nucleocapsid into the cytosol. The viral nucleocapsid or genome are then released directly into the cytoplasm, and the viral envelope remains inserted in the plasma membrane. And the second mechanism of action is receptor-mediated endocytosis, which also called viropexis. The receptor-bounded virus, mainly non-enveloped viruses, are taken inside the cell through clathrin-coated vesicles, which become endosomal vesicles. Now, how the uncoating occurs? Uncoating occurs concomitantly or shortly after internalization. Uncoating is the physical separation of the viral nucleic acid from the capsid or envelope so that it can function. The nucleic acids migrate into the cytoplasm. This migration is direct when most enveloped viruses penetrate the envelope via membrane fusion. But when receptor-mediated endocytosis is the mechanism of entry, hydrophilic structures of capsid proteins may be exposed after viral binding to the cells, and these structures help the virus slip through the direct penetration of the membrane. Cytoplasmic proteases or viral proteases digest the protein layers, releasing the nucleic acid and some proteins that have remained attached to the genome. Now, what happened after the viral genome releasing at the cytoplasm? In this step, synthesis occurs. Synthesis involved replication of the viral nucleic acid and synthesis of viral protein. The essential theme in replication of viral is that specific mRNAs must be transcribed from the viral nucleic acid for successful expression and duplication of genetic information. That means by which various classes of viruses accomplish these steps depends on the structure of genome and the site of replication. Today I am discussing on DNA virus replication, and about RNA virus replication I will discuss on next lecture. In DNA virus also two types: double-stranded DNA virus and single-stranded DNA virus. I am starting with the double-stranded DNA virus. Generally, DNA genome is synthesized in the nucleus of host cell, exception of pox virus. It synthesizes all components in cytoplasm. In early period, uncoating at the nuclear pore, the viral DNA diffuses into the nucleus and transcription by the cellular DNA-dependent DNA polymerase is initiated. At first, only part of the viral genome is transcribed and translated, leading to the synthesis of early proteins which include viral-coded polymerases, regulatory proteins and matrix proteins. In case of regulatory proteins have divergent functions. In classic lytic reaction, the regulatory proteins inhibit host cell macromolecular synthesis, DNA, RNA and protein, and all the metabolic energy of the cell is diverted towards the production of components of new virus particles. The viral-coded DNA polymerase allows the replication of the viral genome, which generate progeny DNA strands. Matrix proteins serve to provide a scaffold for the replication of viral nucleic acid and of viral progeny particles. In electron microscope, matrix proteins look electron dense formation and are called the inclusion bodies. At the late period of double-stranded DNA viruses, during this period, progeny nucleic acid is synthesized by the viral-coded polymerases. Newly replicated genomes also provide new templates that amplify late-gen mRNA synthesis. Late proteins, the structural proteins are needed to assemble the progeny variants. The structural proteins are generally not required before the genome is replicated. Now, what happened in the case of single-stranded DNA viruses? The single-stranded DNA viruses is the parvovirus and contains the DNA sequences that are inverted. They use the cellular DNA polymerases for making viral DNA double-stranded, called the replicative form. Then parental DNA strand is replaced by synthesis of complementary DNA strand to the template strand. Messenger RNAs are synthesized using newly formed dsDNA strand as template. Today's lecture is up to this. I am coming with next video very shortly. So, if you like our video, please click at the like button and share this video in your circle. Don't forget to subscribe our channel. which will help you to gather more knowledges in future. And if you already subscribed our channel, then click at above bell button to know our channel updates. At last, please write your experience regarding this video at comment box, which will help us to evaluate ourselves. We are coming with our next video shortly. Till then, goodbye.